Alright, we have again a nice question here. The linkage map of X chromosome on fruit fly has 66 units with yellow body gene small y at one end. Small y at one end and Bob has small b at the other end. So यहाँ पर है small y और यहाँ पर है small b. The recombination frequency between these two genes, small y and small b, should be 60%, greater than 50%, less or equal to 50%, 100%. अभी you would wonder कि X क्रोमोसोम के अंदर 66 यूनिट्स तो ऑलरेडी है एंड एंड में है स्मॉल y और स्मॉल b, तो डिस्टेंस बिटवीन और रिकॉम्बिनेशन फ्रीक्वेंसी बिटवीन स्मॉल y एंड स्मॉल b शुड बी 66%. 66 centimorgan or 66 map units, which is same as 66%. Then what should be the best option here? वो तो दिया ही नहीं 66%. Of course ये बहुत high level की बात है. They will never ask you this in the NTA paper or NEET paper, but NTA paper में surprisingly दिया. So I'll just teach you this. What happens is this. Please remember, the linear relationship holds true for lower value only. तो अगर यहाँ और यहाँ जीन्स है 1.3% recombination, it holds true. अगर यहाँ और यहाँ जीन्स है 37.2% recombination, जैसे हमने सीखा था ड्रोसोमेला का, then also it is true. But as recombination frequency increases beyond 50%, more than 50%, recombination frequency बढ़ गया 66%, then linear relationship does not hold true due to something called double and multiple crossovers between the non-sister chromatids of homologous chromosome. ये और इसके homologous chromosome के बीच में होता है something called as double crossover or sometimes multiple crossover. So normally एक crossover होना चाहिए, one crossover होना चाहिए, but अगर distance greater than 50% हो, then उनके बीच में multiple crossover या double crossover हो जाता है, and that is the reason recombination frequency is less than the map distance and never ever exceeds more than 50%. 50% से ज्यादा recombination frequency हो ही नहीं सकती है. So what is maximum recombination frequency? 50%, because if it is more than 60% because of double crossover or multiple crossover, the recombination frequency becomes less. So, what will be the answer to the question? Yellow body, small y and bobtail small gene are present 66 map units apart, which means there is less than 50% chance of recombination between them. So the answer is option 3, less or equal to 50%. So 66 units is more than 50% recombination between small y and small b, and because of double crossover or multiple crossover, what will happen? Recombination frequency can never be more than 50%. So though it is 66 units, it is not 66 66% recombination, it is less or equal to 50%. I hope you understood this one. And example 7. The last example which is given in the NTA paper. Drosophila with Y plus W slash small y small w genotype has brown body red eye, yellow body red eye, yellow body white eye and brown body white eye. And what will be the phenotype with this genotype? You may remember right now, brown body and white eye. Brown body and white eye is the answer. ये फिलहाल आपको समझ में नहीं आएगा because there is something called as genic balance theory of sex in Drosophila. Genic balance theory of sex in Drosophila, which is a small variation from how genes pass in Drosophila to decide sex of Drosophila and the coloration of the organs. ये बिल्कुल आपके इंक लेवल में दिया ही नहीं है. Not even in Truman, everywhere it is not given. But I will still teach you this. But in a next lecture, एक lecture वापस मैं Tuesday को आपको देने वाला हूँ. छोटा lecture होगा वो, in which I will teach you about genic balance theory of sex in Drosophila. Chalo, so with that 7th example, we have done with everything. I just want to teach you a small little logic. ये please याद रखना, when two dominant alleles for two different traits, capital A, capital B, are linked on one chromosome, it is called as cis arrangement of genes. तो समझो ये क्रोमोसोम है, ये क्रोमोसोम है, and you have capital A and capital B, which are two dominant alleles for two different traits. A अलग trait के लिए है, B अलग trait के लिए है, and they are on one chromosome. They are on same chromosome, and दूसरे पे है small a small b. Then what is this arrangement called as? Cis arrangement. इसे क्या कहते हैं? Cis arrangement. Cis. And when two dominant alleles for two different traits are present on different chromosomes, and one capital A is on one chromosome and capital B is on another chromosome, but on the homologous chromosome. Then we have capital A and we have small a and we have capital B and we have small b. So what is this arrangement called as? Trans arrangement. This arrangement is called as trans arrangement. So what is capital A and capital B? Two dominant alleles for two different traits are found on same chromosome. Then this is called as cis arrangement. And what if two dominant genes, capital A and capital B, for different traits are present on different chromosomes? Then that is called as trans arrangement. ये एक पॉइंट है, अच्छा पॉइंट है, just याद रखना. You may be asked this in case of the NEET exam. Check. These are the different questions from NTA paper. I hope you have understood completely gene mapping. Now please see the video and understand them. And if you have more questions from the NTA paper, please send them on the doubts group. जितने भी जीन मैपिंग के या आपको कोई भी क्वेश्चन हो, मेरे डाउट ग्रुप पे, विशाल सर के डाउट ग्रुप पे, आर्य सर के डाउट ग्रुप पे, प्लीज सेंड देम. And there are other questions from genetics books These are just for knowledge. If you don't have time, don't study if you don't have time right now. You may pause the lecture right now, stop seeing it. You may not study also the next question that I have given in your PDF file. टाइम है तो एक क्वेश्चन है, सिंपल ही है, देख लो. टाइम नहीं है तो बंद कर लो. क्वेश्चन इन मेल color endosperm capital C is dominant over colorless small c and full endosperm capital R is dominant over shrunken small r. When a dihybrid of F1 generation was test crossed, it produced 4 phenotypes in the following percentage. Colored and full 45%, colored and shrunken 5%, colorless and full 4%, colorless and shrunken 46%. From this data, what would be the distance between the two non-allelic genes? And units are given. Very simple. Color, dominant and full are dominant. Color is dominant, full is dominant. And color dominant, full dominant are parental types, 45%. Colorless, how much is color and full? 45% parental type. And colorless recessive. Colorless is recessive. And shrunken is recessive. So colorless recessive and shrunken recessive are again parental types. So how much is colorless and shrunken? Colorless and shrunken 46%. So how many are the parental types? 45, 46. And colorless recessive and full dominant. So that is recessive dominant are recombinant. And how much is colorless and full? Colorless and full is 4%. So they are recombinant 4%. And colorless recessive and shrunken recessive are, I made a little error here. It should be 5%. Colored and shrunken. I'm so sorry. Just make a little change there. This is colored, dominant, and shrunken. Please make a change in your notes also. Colored, dominant and shrunken recessive are recombinant. And how much is colored and shrunken? Colored and shrunken is again 5%. Colored and shrunken is 5% recombinant. So what are the number of recombinants? Recombinants are 4% and 5%, that is 9%. What is the total number of progeny offsprings? 45% parental, 46% parental, 4% recombinant, 5% recombinant. In other words, 100 is offspring of progeny. So what is the crossover value? Total number of recombinants upon total number of progeny offsprings into 100. So how much is the total number of recombinant? 9. How much is the total number of progeny? 100. Multiplied by 100 is 9% recombination or 9 map units or 9 centimorgans. So what will be the distance between the two or non-allelic genes? 9%. 9% is the answer. So I hope you all have understood gene mapping thoroughly well now. अगर ये आपको आता है, आपको कोई भी न्यूमेरिकल पूछने दो in the NT NEET paper, you will be able to solve it easily. Okay? And एक short lecture मैं आपका लूंगा Tuesday को, which I will send you again on YouTube. That will be some new logic which they have asked in the NTA paper, something called epistaxis, dominant epistaxis, recessive epistaxis. Then also they have asked you about duplicate genes, trans genes, ऐसे करके कुछ नए नए एनटीए पेपर के क्वेश्चन आप लोगों ने मुझे भेजे, सो दैट विल बी half hour 45 minutes maximum. I won't waste your time. Quick lecture I will take on that. I will send you again a PDF file. छोटा सा PDF फाइल होगा, 3-4 पेज का होगा, उसे देख लेना तो आपका वो वाला पार्ट भी स्ट्रॉन्ग हो जाएगा, because they are in the NDA paper. Okay, work hard guys, God bless you, see you very soon on the Zoom session. I have to give you a last minute very important instruction. Please don't miss that Zoom session. You and your parents both have to see that last Zoom session. How to prepare PCB in the last few days and something about how to stay calm and happy in the last few days. बहुत important बातें हैं. I'll just help you out with that. Take care. See ya. God bless you.